Welcome to the latest episode of Star Cells and God. My name is Rana. I'm a biochemist, and I'm joined in studio today by Dr. Hugh Ross. And we're going to be talking about two discoveries that provide scientific evidence for the Christian faith. I'm going to be discussing ancient DNA, and Hugh's going to be talking about bioinspired designs. Hugh, welcome, and why don't you go ahead and lead us off today with your discovery?
Well, we're not talking about the muscles that you have in your bicep. You got big bicep muscles. We're talking about these shellfish, also called barnacles. And I grew up in coastal British Columbia, where people loved to buy boats. Mm-hmm. They refer to buying a boat as the second best feeling a man can have in his life. The best feeling is when you actually sell that boat.
Yeah.
One of the problems about owning a boat in salt water off of British Columbia is that you get these mussels adhering all over your boat. And trying to get those things off your boat, very challenging, just because how strongly these mussels adhere to the boat. You basically are destroying part of your hull to get them off. So it's basically not only you got to remove these things, you got to repair the hull of your boat. And you got to do this about once or twice a year. So after a while you say, I don't want to own this boat anymore. I got to get rid of this boat. As much fun as you might have. And, I've heard people literally saying things, swearing, as they're trying to get these barnacles off their boat. But it just shows you that these creatures, these shellfish, they're tasty when you eat them in restaurants, but they have the property that they can adhere to almost anything. In fact, they have an adhesive that's better than the adhesives that we manufacture.
Yeah.
So that's what we're talking about, a bioinspired design. And so a team of scientists at Rice University basically decided to study these mussels and determine, okay, what's responsible for the incredible adhesive power these mussels have. And they identified an amino acid. You have to be a biochemist to actually pronounce these things. I'm going to give it a go, Fuzz. Please correct me if I don't get it right. It's 3,4-dihydroxyl L-phenylalanine.
Yeah, 3,4-dihydroxyl L-phenylalanine. So there you go.
Yeah, but I prefer the acronym DOPA, D-O-P-A. So I'm just going to call it DOPA from now on. And it's this DOPA that actually explains how these mussels can attach themselves to almost any surface in a way that's virtually impossible to pull them off. And so the team at Rice University said, maybe we can extract this DOPA from the mussels and actually use it to try to take care of a problem we have with plastic pollution. As you're probably aware, we have microplastics and plastics filling up the oceans or lakes and are getting into our food supply, and it's causing health issues, particularly in Asia, where people are throwing away a lot of plastic. And the problem is, even like here in America, we recycle plastic, but it's like if you put them in a dump, it still takes a long time for this stuff to decompose. And when it does, it tends to get into the water system in the underground. So it'd be really nice if we could find a way to take care of this. And the plastic that's of greatest concern is something called PAT, and I'm going to take a stab at trying to actually pronounce what that is. It's polyethylene terephthalate.
Yeah, polyethylene terephthalate. Yep.
Yeah, that's it. It's called PAT. Well, these Rice University scientists found an enzyme that actually decomposes this plastic. And it's a plastic that makes up 64% of all the plastics that's in distribution around the world. So it's the most common and most abundant plastic, but it also has a characteristic. It's the one that decomposes more slowly than any other plastic. Literally takes centuries for PAT to decompose. But they found an enzyme called polyethylene terephthalate hydrolase that decomposes it. And so what they were able to do is that they used a bioengineering technique, and maybe you're familiar with this. They're using what's called genetic code expansion technology to bioengineer bacteria. And so they're using this technology basically to take this DOPA, combine it with this enzyme that decomposes it, and then actually make a protein that they put inside a bacterium.
Yeah.
And the whole idea is that these bacteria were going to be able to attach themselves to microplastics to a degree that's never been seen before. In fact, they did an experiment and they discovered that these bioengineered bacteria are able to adhere to microplastics more than 400 times stronger, more efficiently than anything's been done before. And so their experiments basically demonstrate that if we can set these bacteria loose, these bioengineered bacteria loose in our oceans and rivers and lakes and in our soils, they'll be decomposing this PAT plastic in hours as opposed to centuries.
Yeah.
If you've got enough bacteria, literally in hours, the stuff is gone. So they're saying this could be the way to solve one of the biggest pollution crises facing humanity today. Because it's hard to stop people from throwing away these plastics. And even if you recycle them, you still got the problem. We got to store them somewhere, and it's going to be centuries before this region will be safe again to grow plants or to have our animals walking around in. So the fact that we can get rid of the plastic in hours as opposed to centuries, that would be a huge boost. So I kind of look at this as a creation care thing. But this paper, and it was published in a rather, well, it's a obscure journal to me. It's a journal called Small Methods. I don't know why they chose that title for their journal. But it was a several-page paper where they talked about how we can bioengineer these bacteria, harness them to take care of one of the biggest pollution crises that is affecting humanity. And it's affecting our health. It's affecting our economy. But I liked what it said at the end of the paper. They said, if we just take the protein, it's neat that we were able to put the protein inside the bacterium, but what if we just use the protein all by itself and create a protein film? And what they demonstrated is that you could take medical instruments, like say scalpels or tubes that you put down into your veins. They could actually coat these medical devices with this protein. And what it would do is it would create a barrier. It'd be a film you put all around the medical device, and it'd be a barrier that would prevent any fungi or algae or bacteria getting attached to the medical instrument, which means you could use these devices in surgery and not worry about how you could be infecting the patient. And would also save a lot of time, because a lot of time in the medical industry is going to sterilize the medical equipment. This would basically it'd be safer, more reliable, and so it would cut down the unintended infection rate to a great degree. But they also ended it by saying this would not only be a boon to medical health, it would be a boon to the shipping industry, because what we could do is actually coat the hulls of ships with this protein. All you need is a thin level, but that'd be enough to stop bacteria and fungi and algae from attaching, because it's not just these mussels that'll attach to your ship. A lot of other stuff you don't want attaching to your ship will happen as well. Or think of these undersea pipelines. And one of the problems with pipelines is, you get all this biological material adhering to it, and it starts to decay the pipe. And they said, instead we could use this protein to coat these things. And now we have the assurance that these undersea cables, and not just undersea cables, I know one thing they're trying to do is, instead of having these, poles with electrical wires on it, put it underground. Well, if you put it underground, you want to make sure that those underground wires are protected from biological decay. And so this protein could be used to do that. So multiple applications that would really boost our economy, but also ensure our health and take care of the pollution crises we're facing, especially overseas. So that's the bottom line.
Well, it's interesting is one of the ideas in recycling plastics is to actually turn the plastic back into the monomers that people use to synthesize the plastic to begin with. And polyethylene terephthalate could easily be done in principle just simply through an acid hydrolysis reaction. The problem is polyethylene terephthalate is the polymeric strands are so tightly packed, you can't get acid close enough to do an effective digestion job. So with this type of with this bacteria, you could actually, even before the plastic gets into the environment, take it to a recycling facility and convert it back into the monomers that could then be used to make more polyethylene terephthalate. So you have a stable supply of the monomer and the polymer without ever having the pollution in the environment.
So what you're suggesting, Fuzz, not only set these bacteria loose in the oceans and rivers and lakes, put them in the recycling centers where you got concentrations of this plastic, and that would actually make a lot cheaper to get the plastics back to where we want them in the first place.
Yeah, it's really neat stuff. And and I think the point you were making when we were chatting is, that God has given us everything that we need within the creation to fulfill the mandates that we have as image bearers, one of which is to be caretakers of the planet and to promote human flourishing. And it's interesting because these are not in opposition to each other. What we do to care for the planet actually promotes human flourishing, ironically. A lot of times I think people think that if we care for the environment, it's always at the expense of human flourishing. But that's not the case at all, because the pollution in the environment is devastating to human health.
Right. And again, going back to my youth when I wasn't yet a Christian, hearing all these guys who own boats cursing these mussels, it's like, well, wait a minute. If God created everything and has set us up as stewards, there has to be some reason why he made mussels and made them with such incredibly powerful adhesive forces. Let's see what benefit we can get out of it.
Yeah.
So don't curse anything that God creates. There's a purpose, there's a reason for it. And as stewards of the planet, we need to find the good reason why God made these things.
Yeah.
And often I see that happening when people look at parasites and say, why would a good God do that? But literally every parasite on the planet serves a good purpose.
Yeah.
And these mussels likewise serve a good purpose. And I think this adhesive is going to find lots of applications beyond just what's been discussed in this paper.
Yeah. All right. Well, let's move on, and I'll talk about the discovery that I want to focus on today. But before I do that, I'm going to tell you a little story that will kind of set the stage here. And this is a picture of my dad, and my wife took a photo of him and put it in some kind of metal contraption that sits on my desk. And this is a picture of him when he was a professor at West Virginia Institute of Technology. He was the chairman of the physics department. And my father was an interesting man. He was one of the brightest people I've ever known.
Thank you for showing this photo, Fuzz, because I can see. I look at your features and look at your dad, because you're now almost the same age he was in that photo, and wow, I can tell he's your dad.
Yeah, well, he looks a lot like my brother too. So that's what I thought of when I saw the photo. But anyway, he was a very bright person, and he was a no-nonsense type of guy. He just didn't suffer fools very well. And he was an intimidating person in the classroom. I heard stories from his former students when they learned that I'm, my father's son. They almost always, at least it's been a while since I've met people that were my students of my dad, but they would tell me the same story, how frightening he was in the classroom. He was just a very intimidating person. But what's interesting is that my dad loved soap operas.
Oh.
So here is this
physicist that loves soap operas?
Right.
Wow.
No nonsense guy, but apparently the allure of a good drama and romance was, all it took for my father to get hooked. And so he literally would adjust his teaching schedule so that he could come home to watch two of his favorite soap operas. We lived near the WVIT campus. Then he would go back to class, and spend the rest of the afternoon there. But the point is that, it's hard to turn away from a good soap opera. And if there's a scientific version of a soap opera, it would be the question of whether humans and Neanderthals interbred, right? Everybody seems to be fascinated with that question, regardless of who you are, whether you're...
So that's going to be the next Netflix series?
Yeah, it could be. Whether you're, a scientist or just a layperson, everybody seems to want to know about human Neanderthal interbreeding. And the only reason we know that humans and Neanderthals interbred is because of a major advance that's happened in paleontology that has given birth to a whole discipline called paleogenomics, where we can isolate DNA from fossil remains and actually sequence the DNA, and from that learn about the organisms, the past organisms' biology and their relationship to organisms that existed with them, and then also organisms that exist today. And we've got the, high-quality Neanderthal genomes available to us, high-quality Denisovan genomes. But these are not the only two organisms that we have genomic data from. And this is a figure taken from a paper published in 2023, giving a summary of all the different types of ancient DNA specimens that we have. The brown colors refer to DNA recovered from sediment that we've been able to sequence and learn about microorganisms in the environment, as well as other organisms that would have left behind DNA residues. And there's also the blue circles are hominins that have given up DNA. Most of them are in the 40 to 50,000 year range, but there are some hominin remains that are as old as 100,000 years. And then there's also a number of animals that have given up DNA. The record right now is DNA recovered from a specimen that's about 1.2 million years in age. And this is DNA that is actually sequenceable, right, where we can get sequence data from it. There is suggestion of DNA remnants being recovered from fossils that are 75 million years old, dinosaur specimens. This is contentious, and these are not going to be sequenceable pieces of DNA.
So they're very short pieces?
Yeah, very short pieces of DNA that have been detected indirectly.
Now, there's one at 1.2 million. It looks like it's a mammoth I see there.
Yeah, I think that's right. Yeah, I think...
and how long of a sequence do we have there at 1.2 million?
I don't know the size of the sequences. The rule of thumb would be the older the specimen, the greater the degree of fragmentation, therefore the shorter the sequences. But it's the age is not a good predictor. Probably temperature and other markers of preservation. Really well-preserved specimens that are preserved at cold temperatures can yield DNA of large enough size that you can sequence it, and then through shotgun sequencing techniques and the computational power we have, by comparing those sequences to reference genomes of what we believe to be related animals, can help reconstruct significant portions of the genome.
And that mammoth was probably dug up in some frozen tundra.
Yes.
So chances are it's better preserved.
Most likely.
Yeah.
But the question you're asking is really the question that a lot of scientists are asking, is what is the extent of survivability of DNA in fossil specimens? We have an idea of how DNA breaks down, right? And the thought is that, and this slide summarizes that, is that once an organism dies, there's a very rapid decay of DNA through the action of what are called nucleases, which are enzymes that start chewing up the DNA that are present in the organism. Microorganisms from the environment are going to also facilitate that breakdown. And there's a number of chemical processes that will destroy the DNA. Oxidation, there's cross-linking reactions that will destroy the DNA but will actually preserve the modified versions of the DNA for long periods of time. There's something called a deamidation reaction that converts cytosine to uracil and methylcytosine to thymine. And that's how we actually know that DNA samples are authentic when we recover them from fossil remains is you look for that kind of characteristic damage. And then there's a reaction that's considered to be the most important reaction in terms of destroying the DNA, and this is called a depurination reaction. And just for the sake of completeness, it's an acid-catalyzed reaction, which is an important point, where the proton from the acid attaches to the purine, whether it's adenine or guanine, and that causes a set of domino effects that causes ultimately the DNA chain to break apart, right? And that fragmentation is believed to be the dominant mechanism for DNA breakdown. But the question then is, how long can DNA survive, right? And scientists want to know this because if we know how long DNA can survive and under what conditions promote its survivability, then we can determine, is this DNA that we're extracting from a fossil remain authentic, right? Or conversely, it gives us an idea where to look, right? There's going to be certain specimens we can rule out. Other specimens we want to go through the effort of trying to extract the DNA because it meets criteria. So understanding DNA breakdown is really very critical to the whole area of paleogenomics. Now, what's interesting is young Earth creationists, have latched on to the recovery of DNA from fossil remains and have made the argument, look, DNA is a fragile molecule. It's true. We don't expect it to survive very long. And therefore, the fact that we see DNA from these fossil remains means those fossil remains can't be tens of thousands or 100,000s or even 1,000,000 years old. They must be only a few thousand years old. Therefore, radiometric dating is unreliable, and the Earth is actually young, and the fossil record is best explained by a global flood. This has become a common argument.
I get it all the time on my social media and Twitter, Fuzz.
Yes.
So.
Well, and me too. And a few weeks ago, a young Earth creationist challenged me on this point and cited a paper published in 2012 that shows that the half-life of DNA, according to his interpretation of the paper, is 520 years, meaning that if you use the rule of 10 as, for...
After 5,000 years, it's gone.
So, yes, yeah. After 5,000 years, it's gone. And this paper is a paper that I've seen cited quite often by young Earth creationists making the very point. So this is not something that this young Earth, advocate was uniquely pointing out. I've seen people.
I've seen it on several young Earth websites.
So as I was interacting with him, I told him I would read the paper, and after I read the paper, I thought, this is really worth discussing here on Star Cells and God. Even though it's a 2012 paper, it's still often cited as evidence that DNA shouldn't survive in fossils, at least by young Earth creationists. Nobody who is a mainstream scientist is making that assertion, right? Everybody recognizes radiometric dating is reliable and that this is really bona fide, authentic DNA we're recovering from the fossils. Now let's try to understand mechanistically how it can survive. But because it was a 2012 paper, I also did a little bit more literature work and found a paper published in 2017, as well as a paper published in 2023 that all look at mechanisms of DNA survivability. But again, in this paper, in the abstract, what this research team is doing is they had 158 samples of bones from extinct moa, the New Zealand moa.
Right.
And they had radiometric dates for all of these. So they were able to actually construct kinetic curves showing the breakdown. And one of the questions they had was, is this depurination reaction the primary means for DNA fragmentation? And they were able to show, yes, it was. But what they were doing in their study is they were looking at a 242 base pair region of the mitochondrial DNA, and the mitochondrial genome is about 17,000 base pairs. So this is a small fraction of the mitochondrial genome that corresponds to a control region. So they chose this fragment because this is the type of thing people are looking for in ancient DNA studies, is sections of the genome that would be informative, biologically and phylogenetically informative, right? And so they measured the half-life of that fragment at 520 years. It wasn't the half-life of DNA in the samples, it was the half-life of that fragment. So the fragment could disappear in 5,000 years, meaning you're not going to recover the fragment, but you still can recover DNA that's actually long enough to sequence it. So right away, this person is, and other young Earth creationists are misreading the paper. They're either only reading the abstract and not carefully, and they haven't read the paper, or if they've read the paper, they don't understand what they're reading, or sadly, they might even be misrepresenting the work to, scientifically uninformed people. But the bottom line is, the half-life of DNA isn't 520 years. The fragment recovered from the fossils. And the important point in this paper that we should be paying attention to is the nucleotide fragmentation rate. What is the likelihood of a particular bond being broken in DNA? And they measure that rate at, let's see here, 5.5 times 10 to the minus 6 nucleotides per year. So if you use that nucleotide fragmentation rate and you apply it to a fragment that's 250 base pairs in size at the temperatures that they were working with in their study, which was 13.1 degrees centigrade, which is about 55 degrees Fahrenheit, that fragment would disappear entirely after 181,000 years. So you're going to expect some DNA to be present for about 180,000 years. They also showed in the paper...
That's just in that one fragment.
That's that one fragment.
Yeah.
If you multiply that across the entire mitochondrial genome, that's 13 million years before the genome disappears. Right, so the bottom line is this paper isn't showing that DNA shouldn't be around 5,000 years after the death of the organism. This paper is actually showing that DNA can survive for quite long periods of time. And in fact, they actually show that at minus 5 degrees centigrade, and this is important because the rate of a chemical reaction as a rule of thumb doubles every 10 degrees centigrade. So if you're going from 13 degrees to minus 5, you're going to have the reaction and then have it again.
Right.
And they estimate the half-life of that DNA fragment, at being 158,000 years at minus 5.
At minus 5.
5 degrees.
Just for the fragment.
Just for the fragment. And they expect that you're going to easily have fragments of DNA that are 30 base pairs in size, which are sequenceable, for vast periods of time. And so this is what the last couple of paragraphs in the paper, and I just want to read it again, just to drive home the point that these researchers who've done the work are concluding something very differently than young Earth creationists. They say, it is tempting to suggest that we can now predict the temporal limits of DNA survival and finally refute the claims of authentic DNA from Cretaceous and Miocene specimens. This is, however, not straightforward. One needs information on the number of template molecules in living tissues, estimates of post-mortem DNA decay rates for each tissue type. However, the half-life prediction displays the extreme improbability that an authentic 174 base pair long fragment of 80 to 85 million year old bones could have been amplified. So they question extremely old DNA specimens that are relatively long in terms of the base pair size. Our results indicate that short fragments of DNA could be present for a very long time at minus 5. The model predicts a half-life of 158,000 years for a 30 base pair fragment. So I made a mistake. It wasn't for the 242 base pair fragment. It was for a 30 base pair fragment. So I made the mistake there. And even rough estimates such as this imply that sequenceable bone DNA fragments may still be present more than 1 million years after deposition in deep frozen environments. It therefore seems reasonable to suggest that future research may identify authentic DNA that is significantly older than the current record of approximately 450 to 800,000 years from Greenlandic ice cores. Now, real quickly, I found another paper published in 2017 that makes reference to this paper, and in this paper they look at a number of different factors that are controlling DNA degradation. And here it's a similar type of study where they've got a large collection of fossil remains that they extract, that DNA has been extracted from. The dates of these remains are well known, and they start looking at different factors that seem to control decay rates. And they discover here that age isn't a chief predictor of survivability. Temperature is very important. The association of bone is important for two reasons. One, bone creates a basic or an alkaline environment, not an acidic environment, which means it's going to slow down the fragmentation reaction. Also, the absorption of DNA to bone preserves its survivability and prevents fragmentation, right? And so there's a number of different factors they have identified that contribute to it. One of them, which is very interesting, is diffusion of the DNA away from the sample itself. So they actually argue that the earlier study, which had a half-life of 520 years for that fragment, may have been not due to fragmentation, but due to diffusion of the DNA away from the sample, as opposed to actually breakdown.
A couple more factors, Fuzz. If the bone is, say, embedded in ice, that's, say, minus 30 degrees centigrade, would that cut off liquid water and oxygen? Because I imagine liquid water and oxygen are also factors that would enhance the decay process.
Yes, exactly.
And if that's cut off.
Yeah, and so they expect that you're going to see very rapid decay initially of DNA that then is going to level out, and the DNA is going to be preserved because of these other factors that influence it. Now, there's going to be some fossil specimens that are poorly preserved that are not going to give DNA, but those that are well preserved and that meet certain criteria, cold temperatures, as you're pointing out, where it looks as if there's a very good preservation of the remains. These are all things that are going to contribute.
The bone is not cracked if it's actually encased.
Yes.
No radiation, no oxygen, no water, no bacteria.
Right. So the bottom line is that this is a really, on the surface, it's a very compelling argument for a young Earth. But as you start digging beneath the surface just even a little bit, you quickly realize that scientists are not befuddled by the fact that DNA is surviving, and that there's no reason to question the dates of these specimens. These sample, we understand mechanistically how DNA is breaking down. We're developing a really good understanding of what are the environmental conditions that control DNA survivability. It all makes sense. And so recovery of ancient DNA is a boon to science in that it gives us insight into the biology of the organism. It gives us insight into its relationship with other organisms. We can learn things like humans and Neanderthals were interbreeding because of ancient DNA, which really gives us an incredible appreciation of life, life on Earth in Earth's past.
A stronger refutation of the young Earth argument is that the paper before this, 158 samples, all different dates, that should enable you to develop kind of a decay curve.
Yes.
And the decay curve is consistent with an old Earth interpretation and contradicts a young Earth interpretation. That's a very strong scientific point.
Yeah.
I assume that's in the paper.
Yeah, they did a decay curve, and that's how they determined that the half-life was 520 years for that particular sample. But again, you can't just look at that in isolation of the total study that was done.
Right.
Right. And they do quite a bit of work looking at the temperature dependency of the decay rates, which is where they recognize that the half-life for a 30 base pair fragment is 158,000 years, which means, you're going to get, if you multiply that by 10, that's 1.5 million is at very cold temperatures for DNA fragments that are 30 base pairs.
Well, what
in size.
impressed me, Fuzz, is when you said a 10 degrees centigrade drop, doubles the half-life of the decay of the DNA. So that means if you had, say, 40 degrees, that's a factor of 64 times.
Yeah.
It's longer.
Right.
So, and I noticed that you started off by showing that mammoth, and we were able to get,
right,
actually reproducible DNA fragments that's 1.2 million years old. And I'm thinking, hey, with a factor of 64, we should be able to push that back even farther than 1.2 million years.
Yeah.
And therefore, I'm excited about the idea. Let's go to some place where we know the bones of these animals have been kept under extremely cold conditions, cut off from any water or any radiation or oxygen, and therefore we might even be able to go back, say, 5-6 million years instead of 1.2 million years.
Yeah. Well, and again, this claim of DNA being recovered from dinosaurs that are 75 million years in age, this specimen was an extremely well-preserved specimen, and there were remnants of soft tissues, including osteoclasts, which are these bone cells. And what they did is they stained the nucleus with a dye, or they stained the specimen with a dye, and the dye all accumulated in the nucleus, which is where you'd expect it if it's binding to DNA. But this is, again, could be a relatively short piece of DNA that's undergone cross-linking reactions and is just really a derivative of DNA. And as long as that dye can bind to that sample, it's going to light up as it's a DNA detecting dye. It's going to light up, but it doesn't mean that what you have there is sequenceable DNA whatsoever. So we also want to be very careful about what we're describing as being reported. When people say there was DNA detected at 75 million years ago, that's really shorthand that they're using to say, well, this is something that was chemically derived from DNA that's still remaining at very low levels in the fossil, low enough that we can, or at least high enough levels that we can detect it with a dye. But it doesn't take a whole lot of material to be detected by a dye. It can detect very low levels.
But it does show that DNA goes back at least that far.
Right. Now, this is a controversial claim, though, right, because of the idea that this would be an extreme preservation. But it's not something that the researchers who made this discovery think is going to be routine. This very well could be an oddity, and it may be that this dye was binding to something else other than DNA, and so it may not be an authentic
in terms
detection.
Of responding to these young Earth claims, the fact that you got DNA that's sequenceable back to 1.2 million years, that by itself is enough to make the case.
Right.
And how do you refute that?
Right.
So
well, and the thing is, one way that people have historically or tested for authenticity of DNA recovered from these specimens is looking at the fragment size. So if you've got a Neanderthal specimen that's 50,000 years old, you're looking for fragment sizes that are relatively consistent in size, right? So, you're also looking for characteristic DNA damage, right? And so if you see those, you can be much more confident that this is actually authentic DNA. Because if it's coming from humans that are handling it as contamination or contamination from the environment, these are going to be much larger pieces of DNA, and they're not going to show damage if it's from recent microbial contamination.
Right.
Right. And so, right away that's telling you that this is probably authentic, right? And a lot of times people are going after mitochondrial DNA and there's very high levels of mitochondrial DNA in a cell relative to nuclear DNA. So.
Well, I'm also thinking about what you showed in that first graph. You know, there's a quest to see if, hey, can we actually recover the woolly mammoth?
Yeah.
You know, actually get enough information in the DNA that we could alter the egg of an elephant and have an elephant actually give birth to a woolly mammoth.
Yeah.
But you need really good DNA from these samples in order for that to be possible.
Yeah.
And when I'm kind of looking at this saying, well, we actually look for DNA from woolly mammoths that's been buried in very cold climates that have been undisturbed, we may be able to get high enough quality DNA to make this relatively straightforward.
Well, and even if you're getting relatively short fragments, as long as they're sequenceable, you know, and you're generating large numbers of sequences of these short fragments, you can, by using an Asian elephant reference genome, you can map the sequence.
Right.
Right, and that's what people are doing is, you know, not getting the entire genome that's fully intact, but they're getting fragments that they're sequencing. And then you can use CRISPR gene editing to alter, you know, the genome of an elephant, Asian elephant, to make it woolly mammoth-like.
Right.
Yeah.
Or at least close.
Yeah, yeah.
Okay.
Yeah,
well, this
so,
is exciting.
Yeah, so I'm all done here. So.
Okay.
All right, well, thank you so much for joining us today on Star Cells and God. We'd love for you to join the discussion in the comments below. And remember to like this video and to subscribe for more content. New episodes of Star Cells and God release each Wednesday and are available for you here on YouTube and your favorite podcast app. Be sure to share this video with a friend, and remember, the more we know about science, the more we have reasons to believe.